video lecture on biotechnology. This would have been the class that would have been held on the 22nd, Friday, 22nd of February. And we're going to finish up chapter 5 and start chapter 6. So I'm talking about microbial diagnostics. In our previous slide, we talked about genetic methods for diagnosis of microbial disease. Another way to understand how a host responds to a pathogen is a gene chip. And gene chips have been used to look at the upregulation and downregulation of different genes, whether the genes are turned on or turned off under different circumstances. And Affymetrix, which is a company that makes gene chips, has developed a SARS-CoV gene chip, which can be used to detect SARS, which is severe acute respiratory syndrome. And it basically has probes of the entire viral genome. The SARS virus is quite large, so it contains 30,000 probes, and that can be used to detect the pathogen directly. Other chips can look at host responses to different pathogens. Here is a microbial diagnostic chip that looks at the mouse response to Neisseria meningitidis, which causes meningitis, one of the forms of meningitis, SARS, and then also E. coli. And you can see the different genes that light up. The way this lights up, if it were in color, upregulated genes would be in green, downregulated genes would be in red. Okay, now we're going to finish up this chapter in talking about bioterrorism. And this is an important part of microbial biotechnology because bioterror agents of microbial origin are quite common. If you look at bioterrorism in the whole, as a whole, it's very easy to transport something that can be turned into spores, like anthrax. Bacillus anthracis can be turned into spores. It's a spore-forming bacterium. And so these are very conveniently carried aloft. They were used in the first Gulf War. They were used in the Iraq War. And they were used in a scare in 2001 in the United States, where anthrax spores were sent through the mail to different media outlets and also different congressional offices. And so we need to be careful regarding this, combat this. And these are delivered as aerosols. They can be delivered in crop duster planes. And when you're looking at crop bioterrorism, this is more what we call agricultural terrorism or agri-terrorism. You're looking at something that would decimate crops and would alter the food supply. And then directly in water supplies, there have been instances where things like salad bars have been contaminated with E. coli. Intentionally in bioterrorist efforts. There was actually one up in the Dalles, Oregon, and a case where a group of individuals went to all of the restaurants in the town that had a salad bar, and they contaminated every salad bar. Okay, so these are, it's a very easy proposition. It's very easy to contaminate something with a bioterror agent, so we have to be hypervigilant. Okay. See, there's the different biowarfare agents. Brucella causes brucellosis, and it is a zoonotic disease, so it affects animals and humans. Okay. Bacillus anthracis, which is anthrax. Skin anthrax is not lethal, however, inhaled anthrax is. Clostridium botulinum, which you would find in food poisoning. And these toxins are paralytic, so you actually get some sort of paralysis from them. Ebola virus or Marburg virus are quite virulent diseases. These are both hemorrhagic fevers, so you start to hemorrhage from your orifices. Francisella tularensis, which causes a disease called tularemia. Influenza virus, rickettsia, which is like Rocky Mountain spotted fever or typhus, variola, which is smallpox. And even though smallpox is basically not around anymore, there are countries that still have the virus and could still potentially use this as a bioterror agent. And then Yersinia pestis, which is plague, bubonic plague, and that's a bacterium and is used quite often as a bioterror agent. The other thing you have to think about with these bioterror agents is that they can be genetically modified to become even more virulent, or be genetically modified to be able to form spores. In terms of agroterrorism, here are some diseases for targeting livestock, for targeting plants, and for zoonotic diseases. Livestock foot and mouth disease, swine fever virus, different diseases in plants, which are primarily bacterial or fungal, with some bacterial, and these can decimate crops very quickly. And then zoonotic diseases, which can start in animals and then can move to humans. Okay, in terms of how we can use biotechnology to combat these bioweapons, there's a huge initiative for developing air and waterborne pathogen testing. So, if you have an air sampler that can test, say, a large crowded area like a mall or a stadium, that can periodically test for bioterror agents, the presence of bioterror agents, then that can give an early warning system. Some involve ELISA, and ELISA is enzyme-linked immunosorbent assay. It's a type of identification of specific agents, and this has been used historically. And then with the advent of genetically modified bioterror agents, we need more sensitive and accurate biosensors. So this would be like PCR or even protein microarrays. Okay. And here's an antibody chip. This is actually a protein chip. This would be used for diagnosis of a bioterror agent after somebody has become sick. Okay, so you'd have an antibody chip, and on each square of the antibody chip, you would have an antibody for different proteins, like a protein in anthrax, a protein in smallpox, a protein in influenza. And you would go ahead and you would put blood, probably serum, directly on the antibody chip. Okay, and then you would do a labeling antibody, a sandwich antibody assay, and then you would see which one of these squares and see a characteristic pattern for the pathogen that's being detected. So this antibody labeling chip, or antibody labeled chip, then can be used to diagnose disease. And at this point, I'm going to give you a paper. I'm going to give you an assignment. Oh my goodness, I'm so sorry. So I want you to look up the bioterror agent Mycobacterium fermentans incognitus. It's got 3 names. It's got a genus, a species, and a strain, and its use in the Gulf War. I want you to write a brief paper. It should be descriptive. It should tell me what Mycobacterium fermentans incognitus is, how it was made, who made it, and then how it was used in the Gulf War. Okay, so that's your assignment. I'll go over that again today in class. Okay, and that concludes chapter 5. I'm going to pause this so we can go on to chapter 6. And next we have plant biotechnology, and I would have you pay close attention to this particular PowerPoint lecture because I am a plant biotechnologist. And you know us plant biotechnologists, we're going to ask a lot of questions on the test about plant biotechnology. So what we'll be talking about is plant transgenics and how that comes into agriculture. We'll talk about the different methods used to transform plants. There are lots of different methods, and a lot of them were blocked from use by the general public because of patents. And so there are alternative methods to work around those patents. We'll talk about some of the practical applications of these methods. And then we'll talk about health and environmental concerns. And I want to spend a lot of time on this. It's very important. The GMO debate is an extremely important debate. We need to have these conversations regarding genetically modified organisms. And so I want to spend a lot of time on that. Okay, so plant transgenics. And if we look at how the world population has grown, it is doubled in the past 40 years, but the amount of arable land is only increased by 10%. So we have to feed all these people. Now, in terms of population growth dynamics, it actually turns out the world population is now becoming stable at about 7.6 billion. And so some of these arguments may not last very long. Certainly I am not speaking out in favor of world hunger, but as we are able to better use land and better control population, then we may also look at safety of these plants and not trading our ability to feed people for perhaps lack of safe plants. Okay, so there's my little commentary. Now, you can improve crops through traditional breeding methods, and it's allowed us to expand crop variety. It's also allowed us to increase crop productivity, but arguably crop breeding is slow and it probably cannot keep pace with population. Okay. And so one of the big ifs is, will genetic modification of crops increase their productivity? So we have to take a look at that. Okay. Now, if you look at the advent of GM crops started in 1996. They were first approved for growth in the United States in 1996. I was a part of that revolution. I started genetically modifying plants myself in 1994. And so we had plants ready to go, and ours were not grown out in the open field, but they were grown in greenhouses. So we were very, very excited about this. And now if you look at even as late as 2009, then there's over 2 billion acres of biotech crops. These are genetically modifying crops growing worldwide. So it's a sizable portion of all of arable land. Okay. So plant transgenesis or plant transgenics is a direct transfer of genes to plants. Okay, you can actually develop plant vaccines, and that's odd. We need to talk about that because plants don't have an immune system, but they do have defense mechanisms. You can then develop plants that produce their own pesticides. Those are called BT plants, and you can produce plants that are resistant to herbicide. The most common type of plant that's resistant to herbicide we refer to as a Roundup Ready plant. Okay, and by 2008, 13.3 million farmers in 25 countries were planting transgenic crops. They do it here locally. Okay. And if you look at worldwide how this has impacted our different crop plants, 70% of the soybeans worldwide are transgenic, 40% of all the corn, and 10% of all the cotton. In the United States, it's more like 90% of all the soybeans and 70% of all the corn are genetically modified. And you find soybeans and corn in a lot of processed food. You may not know you're eating soybeans or corn, but they have a lot of protein, they have a lot of fillers, they have a lot of starch. And so it's, you think of high fructose corn syrup that comes from genetically modified corn. You're drinking it in a sweetened soda. And so it makes its way into a lot of products that you wouldn't associate with corn or soybeans. Okay. It's also the focus of considerable controversy. Okay, so let's look at how we conventionally breed plants and how you produce hybrid plants or hybridization. Okay. I don't understand this slide. I have to take a step back. Now, I guess we're comparing conventional selective breeding and hybridization to genetically engineering techniques. Now, I guess to hybridize a plant using genetic engineering techniques, you can do what's called protoplast fusion. And say if you have two types of plant cells, say you had a carrot cell and a beet cell, you could actually strip off the cell walls using an enzyme called cellulase, and that would leave the remaining cell, which we call a protoplast. Then you take the protoplast and you mix it with a polymer called polyethylene glycol. That opens up the cell membranes and it causes two protoplasts from two different plants to fuse together. So all of a sudden you have a large protoplast. It has all the chromosomes for beet, it has all the chromosomes for carrots, and then you see what it produces. You actually regenerate the cells and then regenerate entire plants in that way. Okay. The easiest genetic engineering technique I've talked about before is the Agrobacterium method. That's where we use the Thai plasmid. And the Thai plasmid is just naturally resident in Agrobacterium. In the wild, Agrobacterium causes TI, or tumor induction. Okay, TI is tumor induction. And a portion of the plasmid, or the Thai plasmid, is transferred to the plant, and that has virulence genes that causes tumors. Okay. In the Agrobacterium method, we just basically replace the virulence genes with genes that we're interested in, like something that would make insulin. And we can put that in the Thai region, and Agrobacterium will automatically transfer that gene to the chromosome of plants. There are other contraptions called gene guns, and the gene guns have gold bullets, and coated on the gold bullets are pieces of DNA, and the pieces of DNA are aimed towards the nucleus of the plant cells, and they're fired directly towards the nucleus, and that way the DNA is delivered into the nucleus and it's taken up by the chromosomes. You can also do what's called chloroplast engineering. You have to remember chloroplasts have their own genome, and there are lots of individual chloroplasts in each plant cell, and so you can take a small ring of DNA or a small portion of DNA and insert it into the chloroplast genome. There's also what's called antisense technology, and this is just like interfering RNA or RNAi, where you introduce a small amount of genetic material. That genetic material then will block a particular protein from being produced by the plant, and that's been used, for example, slow ripening technology. There's a type of tomato that ripens very slowly and ripens on the vine and then stays ripe without rotting. It's called a flavor saver tomato, and the genes associated with the plant rotting and ethylene production have been downregulated using antisense. Okay. Now, if we take a step back, and now we'll go back to conventional selective breeding and hybridization. I apologize for the confusion in the previous slide. What you do is you take two similarly related plants and you do a sexual cross between those plants. You take the flower portion, the pistil and the stamen of one plant, and then you mix the pollen from another plant, and you come up with a hybrid. And then in order to get the trace in, you repeatedly backcross with the original plant parents until you get the trait that is desired. This can take years because you can't really sexually cross a plant until it's sexually mature. So you have to wait until the plant reaches sexual maturity. Once it reaches maturity, then you can do a sexual backcross. So it takes a lot of time between successive generations. Okay. The thing that's also difficult about selective breeding and hybridization is ploidy. Okay, you may remember the term ploidy from genetics. We are diploid, humans are diploid, but a lot of plants are polyploid. And not only can they carry as much as 4 copies of each chromosome, like potato, but in some cases they can carry 8 copies of each chromosome. So since they're not diploid like us, then it can take a very, very long time before you have a genetic trait that is stable in every set of chromosomes. Okay. Okay, so let's go back to protoplast fusion. Take a look at this. Okay. Protoplast cell is actually a callus cell, and a callus is a cell that has had its cell wall dissolved by the enzyme cellulase. Cellulase just breaks down cellulose, which is in the plant cell wall. And a callus cell is usually grown in the presence of hormones. It's a type of plant cell that is dedifferentiated. A lot of times it's not even green. It doesn't really have that much chlorophyll in it. And in the presence of hormones, it grows very, very quickly. And the cells are crumbly. We call that friable. And when they're friable, they're easier to work with in the laboratory. So we take this callus cell and we dissolve off the cell wall. And then we fuse two protoplasts together. And that is a very simple procedure. You simply take the protoplasts, culture them together, and then add some polyethylene glycol. That causes the cell membranes to merge together, and then that will grow into a hybrid plant. Okay. Broccoflower is a protoplast fusion where they actually took the genome of broccoli and fused it with the genome of cauliflower. So you can expect that it would look a little bit different. It looks like a green cauliflower. Okay, and it has the full genetic complement of broccoli and the full genetic complement of cauliflower, and it grows and divides normally just like any other plant. Okay, and so when you do a protoplast fusion, oh, this is really small. I'm so sorry. Okay, but when you do a protoplast fusion, you take a leaf and you cut it into pieces. You dissolve it with cellulase. Okay, and the cellulase will get rid of the cell wall. Okay, then you centrifuge this to get rid of the debris, and the protoplast without the cell wall will float. Okay, the cells that still have a cell wall will sink. So you recover these, and then you grow the protoplast directly on filter paper. Okay, I would do this a little bit differently. In order to get the protoplast to fuse, you actually want to add polyethylene glycol in this particular step. Okay, here's my arrow, polyethylene glycol. And then you let these grow on growth media, and then you transfer them to shooting media, transfer it to rooting media, and then you get full plants. Okay, so that's how protoplast fusion works. Now, Agrobacterium technique is one of my favorite techniques because it's easy. I like to do easy things and not hard things. Okay, so all you do is you take small discs that are cut from the leaf of tobacco or potato or another type of plant, and then you culture the discs in a medium that contains genetically modified Agrobacterium. The specific species is Agrobacterium tumefaciens. Agrobacterium tumefaciens is a soil bacterium that infects plants. And like we talked about the Thai plasmid before, Thai plasmid can be genetically modified. And the genes that would normally induce a tumor by Agrobacterium are taken out. And the genes that of interest, say if you want to produce like a blood clotting factor in a plant, are put in. Okay, then just the process of co-cultivating the Agrobacterium with the plant allows that Thai plasmid to integrate a portion of itself directly into the chromosomes of the plant cells. Okay. Then once the leaf discs have been transformed, you treat them with plant hormones to stimulate shoot and root development. And I should also mention that Agrobacterium is not a foolproof process. Not every cell is going to be transformed. So we have to use selection pressure here. So we confer antibiotic resistance to the plants, and we grow them up. When we're done treating the leaf disc with Agrobacterium, we grow the leaf discs on selective media that contains an antibiotic called kanamycin. And if it's been successfully transformed, then only those transformed plants will grow in the presence of that antibiotic. Okay. Now, normally what happens with Agrobacterium is that you have a wounded plant. It gets a wound, it's got hacked by some type of mechanism, or maybe it's been bit by an insect. Agrobacteria will infect the wound, and they will produce a tumor called a crown gall. Okay, crown gall just looks like a big bunch of undifferentiated tissue right on the stem of the plant. Okay, what happens is that Agrobacterium has a Thai plasmid in it, and a small region of the Thai plasmid is called tDNA, and that's transferred directly into the chromosomes of the plant cells. tDNA integrates directly into the plant cells, and with wild Agrobacterium, this causes the crown gall tumor. okay? With genetically modified Agrobacterium, where you have a nice benign type plasmid that's not going to cause a tumor, you take the leaf discs, you culture the leaf discs with Agrobacterium for about 24 hours, okay? And then you transfer this to a growth medium, okay? You culture about 2 to 3 days. And I would say, when I was doing this, I would culture this for more like a week. And this shows that there are nurse cells at the bottom, that there are plant cells that are giving nutrients. That's not really viable. That's not really how we do it. Typically, using nurse cells is really easy to contaminate. And so all you do is you take these transformed leaf discs and you put them directly on growth media. So you put them directly on agar that's specifically formulated for rapid growth of plant cells. Okay, this will stimulate growth, and then you put it on shoot induction media. And in shoot induction media, you'll get little shoots that appear, green shoots on the margins of the leaf discs. And then you put them in a plant growth box. So you just put a sterile box and you put them in media that induces roots. And then once the plant is ready to survive on its own, then you move it to soil. Now, in each step, in the initial culture, shoot induction, and root induction, you always have the antibiotic kanamycin, so you make sure that the plants that grow have truly been genetically modified. Okay. Now, Agrobacterium has been the subject of a patent dispute for many, many years. Okay, it makes it a very difficult technology to work with. You have to get a license from a company called Syngenta in order to use Agrobacterium. And so there are alternative technologies, not because they're better, but just because they don't involve that particular patent. And one of the technologies is called the gene gun. Okay. And what this is used to do is you take tiny metal beads, usually they're gold beads, they're coated with DNA, and they're blasted into an embryonic plant cell, usually just the cotyledons from a plant seed. And you take this gene gun, you aim it directly at sterile plant material, and then you bombard it with the gene of interest. Okay? You can aim it at the nucleus, or you can aim it at the chloroplast. If you do a nuclear transformation, then obviously it's going to be in the main chromosomes. If you do it, aim it towards the chloroplast, then it's going to be in the chloroplast genome. Always use a marker gene. Okay, so you have to confer antibiotic resistance. And this is the transformation frequency of the gene gun is quite low. With Agrobacterium, you can get rates of transformation that are very high, such that for every leaf disc, you'll have at least one transformed plant. The rate of gene gun transformation is about 1 in 100. So for every 100 gene gun experiments, you'll probably get one transformed plant. But it's not as satisfying, for sure. Okay, and like I said, people wouldn't do this. They wouldn't go to this level of work, but they have to work around a broad patent on the Agrobacterium method. And that's why this technology was designed. It was designed by DuPont, and DuPont went and patented their technology. Okay, so we have an inferior technology to Agrobacterium, but it was patented as well. Here's a gene gun. Okay, and you just have your plasmid. You coat this on, they show tungsten. I've seen gold used before. You load it, and it's like a little potato gun, okay? Firing pin, you've got a blank charge and the particles, and then you aim them directly towards plants, okay, plant cells, and some of them will enter the nucleus, okay? Some of these particles, they'll deliver the DNA, and then the DNA will be taken up by the chromosomes. Then you grow this up on selective media, and only the selected transformants will grow. Now, another way to do this is chloroplast engineering, and chloroplast engineering is more of a broad term. You can use gene gun in order to engineer a chloroplast. There are other ways to engineer a chloroplast, but you have to figure that there are many, many chloroplasts in plants, and chloroplasts will divide independently of the plant cells. And so you can get a high level of gene expression if you can get a lot of the chloroplasts actually transformed per cell. Okay. The DNA in the chloroplast can actually accept several new genes at a time. So you can get a whole pathway into the DNA of a chloroplast. And a high percentage of the genes will remain active. So you retain activity, and you can do specialized types of transformations because you can insert multiple genes. Now, a nice thing about this is when you grow plants in the wild, genetically modified plants in the wild are going to release genetically modified pollen. And that's a big no-no. You don't want that to happen. You don't want your pollen to drift around and contaminate non-GMO crops. But chloroplasts don't make pollen, okay? That comes from the nuclear genome. So when you have a chloroplast transformation, then the gene of interest is not transferred via the pollen. The gene of interest is transferred in the chloroplast. Okay, so you have no chance that the transformed genes are going to spread via pollen drift. Okay, and you have to imagine that on a windy day with a high pollen producing crop like corn, that pollen is going to go everywhere. So chloroplast engineering safeguards to make sure that you don't have gene drift and you don't have genetically modified traits in non-GM crops, which nobody really wants. Okay, I like this slide because I did it. This is me. Check me out. Okay, here we're taking an antibody. So we have the light chain of an antibody and we have the heavy chain of an antibody. And we do Agrobacterium mediated transfer into tobacco. And so you have a light chain plant and a heavy chain plant. And then you cross the plants and they assemble an entire antibody. Okay, that's the old method. Okay, that's me. I'm old. I'm 55. So that's what I did when I was a younger man. Now, with engineered type plasmids that actually go into, you can have Agrobacterium go into the chloroplast. You can put the genes for the light chain and the heavy chain all on one plasmid. Okay. So you put the light chain in one plasmid, the heavy chain in the same plasmid, you transform the plant so you get chloroplasts that are engineered, and then the chloroplasts are actually expressing the protein. Okay, so you can do this for antibodies. The nice thing about this is you don't have to do two transformations and cross the plants. There's a lot of variability when you cross plants, and you can end up with a lot of low producing plants that don't give you a whole lot of antibody, that waste your time. However, if you put both the light chain and the heavy chain on the same plasmid, then you only have to do one transformation and one set of screening in order to find stable plants that produce lots of antibody. Remember, I would go back to this. Remember the outbreak of Ebola? There were two patients that actually were missionaries that came to the United States, and when they came to the United States, they were infected with Ebola. They were treated with antibodies that were made from plants. There was a company called MAP Pharmaceuticals, run by some friends of mine back from my old plant transgenic days. And the antibodies that they produced were produced in this particular fashion. They came from plants. The antibodies directly bound the Ebola virus, and they were used as a cure. They actually cured these patients of Ebola. So, fun fact. Okay. Another thing that we can do in plant transgenics is antisense technology. Okay, and this has been really big for anti-ripening, to make sure that you can have like a nice vine-ripe tomato, but it's not going to rot during shipping. Okay, so you insert a complementary copy of a gene into a cell. Complementary just means that it's going to be comprised of complementary strand. The gene encodes mRNA, and that mRNA is called antisense. You guys know how antisense works. It just blocks sense and blocks translation. Okay. Antisense RNA binds normal messenger RNA and inactivates it. Okay, you get all these things like slicer and dicer and cutter through RNA interference, and this antisense molecule then will block a translational pathway and block the production of a particular protein. Okay. The big example was a tomato that came out in 1996 called the Flavor Saver tomato. It was a tomato that you could ripen on the vine, you could get it nice and red, but then it wouldn't rot during shipping. Normally, tomatoes are picked off of the vine before they're ripe, and then they're ripened chemically after they've shipped. But with a Flavor Saver tomato, the enzyme that breaks down pectin, polygalacturonidase, or PG, was suppressed using antisense technology, and that suppressed the ripening process so the tomatoes didn't rot. And here what was done, and this is a confusing slide because it looks like PG is a caterpillar, doesn't it? Okay. So in a normal tomato, you have normal messenger RNA. This is sense. That makes polygalacturonidase or onidase. Okay, this ripens the tomato, and then naturally it rots over a period of time. Okay, polygalacturonidase accumulates, it breaks down pectin, and that causes the tomato to rot. Excuse me. So then, if we can take the antisense, then here the sense and the antisense bind. Okay, and you can't translate double-stranded RNA. Bad news, can't translate it. Okay, so when you do this, you just splice in the polygalacturonidase gene into the transgenic tomato. It produces antisense mRNA. Then you get double-stranded mRNA, and that blocks, okay, that blocks translation, and then you don't get polygalacturonidase production. And then the ripe tomato, it will rot eventually. There are other enzymes that will rot a tomato, but it can be vine ripened and then shipped over a period of 3 weeks, and it still will not rot. That's called a Flavor Saver tomato. Other practical applications in the field. Okay, so I've talked about different genetic modification methods, talked about protoplast fusion, we talked about Agrobacterium, we've talked about gene gun, and then we've also talked about antisense technology. Oh, we talked about chloroplast engineering too. Another thing that you can do is you can produce vaccines for plants, which is kind of an odd concept because plants don't have an immune system. Okay, I'll talk about that further. You can create genetic pesticides that are specific to the genomes of different pests. You can create herbicide resistance, which is quite prevalent. Most of the crops that are produced today are herbicide resistant. So you can spray the plant directly with herbicide and all the weeds will die, but the plant will still survive. There are some plants that have been created for enhanced nutrition. And then we'll talk about pharmaceuticals, that's what I did, and we'll talk about fuel production. Okay, let's talk about vaccines for plants. So what you do is you take a vaccine. This is just a DNA for just like a pathogen protein, or say if you want to make the plant resistant to insect attack, you encode the protein for that particular pathogen directly into the plant's DNA. Say if we want to inoculate a plant against tobacco mosaic virus, then we take a gene from tobacco mosaic virus and we insert it into the tobacco plant. And what the plant does is it sees the viral gene, the gene gets expressed, and that stimulates the plant's chemical immunity. Now, the virus in this case doesn't infect the plant because it's just a gene. It's not a whole virus, it's just a gene from the virus, so it's not infective. But because that plant's chemical immunity is stimulated, then when the virus does try to infect the plant, it's impervious, it's invulnerable to the virus, and so therefore the plant is producing specific chemicals that make it resistant to tobacco mosaic virus. Okay. This is tobacco mosaic virus. It's a helical virus, and you just take the DNA for a protein subunit. Okay, just take the coat Actually, this is RNA because tobacco mosaic virus is a RNA virus. Okay, you convert it into DNA, and then you put that onto a plasmid, transform the plant cell, and then when you regenerate the plants, you just add the virus to the leaves, and simply the inoculated plants will live, and the non-inoculated tobacco plants will die. Okay. Genetic pesticides. There is a bacterium called Bacillus thuringiensis. That's difficult to pronounce, but you should know it. Bacillus thuringiensis. BT is its short name, and it produces a protein that will kill harmful insects. And what has been done in the past with the BT bacillus is that it is grown up, the bacterium itself, sprinkled on plants just like a powder, and it's used as an insecticide. Okay? To take this one step further, okay, and this has been used, just they sprinkle the bacteria directly on the plants, and it's been used for over 50 years. Okay, but you can take a toxin gene, a toxin from Bacillus thuringiensis, and you can insert it in the plant's DNA. And what that does is every cell now will produce a toxin that is toxic to insects. And so when the plants are being eaten by insects, the insects ingest the toxins and die. Okay, so the plants are naturally just repel insects. Okay. There are some controversies over this because of the BT gene. The BT gene has not been shown to be safe in humans. And the company that first used the BT gene, Monsanto, claimed that it would be safe in humans, but they claimed that it would actually be destroyed in the gut, okay, in the intestines, in the stomach of humans. It's not. So we have a problem. So we need to face that problem. The other thing is that the gene is actually causing problems with monarch butterflies. And we're starting to see, because of overuse of BT genes in genetically modified plants, the monarch butterfly population is decreasing. I'm going to show some videos about that, how the unintended consequences of genetically modified plants. So we'll take a look at those when we meet today. Okay. So hot button issue. We were using the BT toxin. We use the BT toxin a lot. And there's a lot of genetically modified corn, especially, that has been transformed to produce BT toxin. And in 2011, in reproductive toxicology, scientists looked at pregnant women who had consumed genetically modified corn, and they found that BT toxin was detected in the blood of 93% of the patients and about 80% of the cord blood of pregnant women. What they would do is they would just wait until the pregnant woman gave birth, test the cord blood, and the cord blood then would show the presence or absence of BT. Okay, so this is a big problem. We don't want BT going to developing fetuses. Industry the same year claimed that the toxin would be destroyed in the gut, okay, that humans would eat it, that the toxin would never make it out of the gut, but that wasn't really the case. Okay, and so you have to ask, how did the toxin get there in the first place? Well, number one, you could ingest the plant, and BT toxin is in the plant, so it's going to get into your bloodstream. Okay, the second way that this can happen is called DNA hopping, or horizontal gene transfer. And what happens is, when the DNA is exposed, when you chew food and you expose the DNA and it's being digested in your gut, DNA can hop to gut flora. And this it's called low-level horizontal gene transfer, and transgenes from plants have been shown to hop to your gut bacteria like E. coli. Okay, and so it has been established that plant to bacteria horizontal transfer of genes can occur. Pontaroli in 2009 established this. Okay, it has been denied that this would happen in gut bacteria. Okay, Demonash in 2008 in the Proceedings of the National Academy of Sciences said no, But you have to look at this, and it was a faultily planned experiment, if I do say so myself. The principal investigator, that's what PI means, the principal investigator, his name was Simonette, and he had his own plant biotech company. And so he had a real big conflict of interest, and he had a vested interest in plant DNA not hopping to gut bacteria. So you have to look at that. And if we look at an experiment that was done about 4 years earlier, this was Netherwood in 2004 in Nature Biotechnology. And what they did is they looked at ileostomus. Ileostomus are people that have their ileum is short-circuited out to outside their body, and so they poop in a little bag. They don't poop through their colon and through their rectum. They poop in a little bag. So they were testing ileostomus because ileostomus you can really get at the gut bacteria very easily. It doesn't go through the colon, and it's not adapted through the colon, and a lot of gut bacteria will die in the colon. So they were testing ileostomus, and what they found was that out of 7 ileostomus, 3 of the 7 showed low frequency gene transfer from GM soybean to the microflora of their small bowel. Okay, so what that means is they were actually seeing BT genes in small bowel bacteria, and the genes were being transferred. So they answered their own question, yes. And low-level gene transfer, all of a sudden you've got genetically modified gut flora, and you're like, what do I do? Okay, because your gut flora can start to produce Bt toxin. So that's a really interesting question. Here's Bt up top. It's just a Bacillus thuringiensis. And here is a caterpillar that's attacking a leaf. And Bt, it was very revolutionary, but you have to ask at what cost. I'm going to check the time here really quick. I can go a little longer. Okay. Another thing that is done in terms of practical applications is safe storage of crops. You don't want your crop to rot while it's in storage. Okay. And insect infestations in crops happen during storage. You get weevils in corn and in wheat and in cotton and things like that. So avidin was being expressed in transgenic corn, and that blocks the availability of biotin. And biotin is a type of vitamin that insects need to grow. And so if they express biotin, and the insects would die off. Okay. Another application that's very important is herbicide resistant. And it would be desirable if you could take weed killer like Roundup and you could spray it directly on the crops, because crops get weeds, and to weed crops by hands is very expensive. Okay. So scientists ask, can you genetically engineer crops to be resistant to common herbicides like Roundup? Okay. One example of this resistance is a compound called glyphosate. Okay, so I want you to say glyphosate with me. Glyphosate. Glyphosate is a chemical component of Roundup. Okay, glyphosate blocks a functional enzyme called EPSPS, and that kills a key biochemical pathway in weeds. Okay, it kills a key biochemical pathway in just about any plant. Okay. So crops have been transgenically produced that don't need EPSPS. Okay, so if you have an alternative enzyme that is insensitive to glyphosate, okay, then the plant is going to look at glyphosate and just laugh. It doesn't need it. It's not going to do anything to harm it because it has an alternative enzyme. So get this, we're replacing the enzyme that's sensitive to glyphosate to something that's resistant to glyphosate, and then all of a sudden our genetically modified plant, we can spray Roundup on it all day. It doesn't care. Okay. And most soybeans grown today are herbicide resistance gene. And you might say, Brian, I don't eat soybeans. Yes, you do. If you eat processed foods have a lot of protein additives, and there's a lot of soy in processed food. And unfortunately, glyphosate-resistant weeds have now evolved because of horizontal gene transfer into other plants. There are weeds. It's difficult to stay one step ahead of nature, and so there are glyphosate-resistant weeds, and so you can spray Roundup on the weeds. Same thing happens to them; they just grow. As far as enhanced nutrition, one of the big advances has been golden rice, and golden rice is grown. It is actually golden in color because you take the beta carotene, which is found in carrots, converts in the body to vitamin A, which prevents blindness. And so they've genetically modified the rice so it makes beta carotene. So it looks golden. It looks like a carrot. Okay, it's got the golden color associated with it. This was quite an extraordinary effort. It took over 10 genes being moved into the rice plant. Okay. However, there's a real outcry against GMOs. And as of 2011, even though, this is a crop that I think may have benefit, I'm still kind of on the fence about it, but no farmers have planted golden rice. This is as of 2011, so it's about 8 years old, because of environmental concerns with GMOs. Okay, and I think I will stop there. It's been fun, and I will see you in lecture today.